Hello everyone, welcome back to Food Science. This is the third video on food science of carbohydrates. In this video, we are going to look at the carbohydrate analysis and a little bit about sweetness. Talking about carbohydrate analysis, why it is important to determine the type and concentration of carbohydrates in food. One is for the standards of identity, because the foods must have compositions which conform to government regulations. For nutrition labeling purposes, to detect adulteration. Adulteration is that the food is sometimes mixed with other components, which may be food components or some non-food components. For example, milk, liquid milk, may be mixed with water to increase the volume, or it may be mixed with urea, which will mimic as a protein component. So to make sure that the food is safe and it doesn't have any other things mixed with it, we need to do this carbohydrate analysis. And to determine the quality of the food, and also in food processing, because sometimes the food processing operations that depend on the type and the concentration of carbohydrates that are present. So let's first have a look at some simple tests that can be used to identify the presence of carbohydrates. The first one is the Molish test, which is used to identify all types of carbohydrates. Carbohydrate, in the presence of concentrated sulfuric, forms these furfuryl compounds, which then reacts with alpha-naphtol to form purple or violet colored product. So the appearance of a purple ring confirms the presence of a carbohydrate. The second test is the feeling test, which is used to identify the presence of reducing sugars. So this feelings reagent contains copper as a component. So this reducing sugars with this feelings reagent forms a red precipitate, which confirms the presence of these reducing sugars. The next is the Benedict's tests. This is also used to confirm the presence of reducing sugars, which also forms a red precipitate. The next test is the Tollens test. This Tollens reagent contains silver. So Tollens reagent, it undergoes a series of reactions and it forms silver as a product. This silver deposits in the inner walls of the test tube, which is called the silver mirror, and this confirms the presence of reducing sugars. The next test is the iodine test. Starch reacts with iodine and it undergoes a multi-step process to finally produces a blue color. This dark blue color confirms the presence of starch. So all these tests we described here are simple chemical tests which can be used to identify the presence of carbohydrates. However, these tests are not used presently in industry because the modern food industry uses more sophisticated techniques which are able to accurately and efficiently detect the presence of carbohydrates. So here you can see this picture, which is a food item, and you can see the nutrition information here. So in this information, we can see the numbers representing the content of carbohydrates and also sugars. So let's look at the methods that are actually used in industry to generate these numbers. To get the total carbohydrate component, the most commonly used method is the calculation by difference, which means the total carbohydrate equals to the total weight of the food serving minus the weight of the other compounds, like the weight of crude protein, fat, moisture, and ash. So the industry uses different other techniques to quantify proteins, fat, moisture, and ash, and then they use this this simple equation to get the total carbohydrate content. And then to get the amount of sugars, they use different sophisticated techniques. Let's go through them. The most commonly used are the chromatographic methods. These are the most powerful analytical techniques for the analysis of the type and the concentration of sugars. Techniques like thin layer chromatography, high performance liquid chromatography, HPLC, and gas chromatography are used to separate the carbohydrates, and these techniques are then coupled with other identification techniques like NMR and mass spectrometry, so that they generate peaks, and then we can identify and we can quantify the carbohydrates. The diagram on your left-hand side shows thin layer chromatography. So this thin layer chromatography has several components. One is this TLZ plate, which is a silica plate. This is also known as the stationary phase. So we should have our carbohydrate sample in the form of a solution. So we will first have to put a drop of this sample on this TLZ plate. And then we have the TLZ chamber, which is this beaker at this moment. And this chamber has a solvent. This solvent is known as the mobile phase. And this solvent can be selected based on what components we need to separate. Then we will be introducing this TLZ plate into the chamber. Simply, we are placing this plate inside the chamber. And then this solvent in the chamber will start rising up through this plate. So we will just keep watching while this solvent is moving up. So this solvent is not moving up alone. It's also carrying the components of this sample. So this sample that we introduced at first is now getting separated into its individual components. So we should wait until this solvent reaches the top, and then we can get this TLZ plate out. So we can see that this original sample is now separated into three components: compound A, B, and C. So this high performance liquid chromatography, which you can see on your right hand side, it also uses the same principle. So here we have a solvent at this point, and we will be injecting the sample, which is also in a solution. We'll be injecting this, and then this sample along with the solvent is moving through this HPLC column. While it's moving, it gets separated into its individual components, and then it reaches the detector, and the detector, this is now coupled with other techniques like mass spectrometry to generate this output on a screen. In the screen, we can see different peaks being generated for each component that is being separated. These peaks are generated. So we can read this output. We can compare this output with a standard output to determine what components were there in this original mixture, and also we can quantify their amounts using this technique. So in gas chromatography, similarly, we have this column and we are injecting the sample. However, at this time, we don't have a solvent. Instead, we have a carrier gas. Therefore, this sample, the carbohydrate sample at this point, should be chemically modified into a volatile compound. And this also has a detector which can then identify the different peaks generated, and we can then compare these peaks with a standard to identify and also to quantify the carbohydrate components. So these techniques are the ones that are commonly used in present industry to identify and also quantify the carbohydrates. Now let's talk a little bit about sweeteners. Carbohydrates, including sugars, are used in food industry for a wide variety of applications, such as reducing sweetness, colors like browning, adding flavors, adding calories, giving the texture, thickness, etc. We learned them throughout this lecture. So giving the sweetness is one of the major uses of sugars in food. Glucose, fructose, sucrose are sweet in taste, while fructose is the sweetest sugar. Lactose, which is the sugar present in milk, is the least sweet. According to Australian Bureau of Statistics, it is reported that Australians consume excessive amounts of these sugars. According to the statistics, 46% of the Australian adults exceeded the WHO recommendation of the sugar consumption. And we know that sugar consumption, especially the excessive sugar consumption, is associated with a number of non-communicable diseases, including type 2 diabetes, cardiovascular diseases, and obesity. Therefore, as non-caloric or zero-caloric alternatives to these sugars, artificial sweeteners are introduced. Those are aspartame, saccharin, sucralose, neotame, stevia, etc. Although we consider them as sweet, according to recent literature, the safety of artificial sweeteners also remain controversial because some scientific evidence says that these are also associated with non-communicable diseases like diabetes, cardiovascular diseases, and there are different mechanisms proposed to that. And also some people report that they face gastrointestinal symptoms after consuming these artificial sweeteners. Therefore, we should keep in mind that artificial sweeteners are also not 100% safe according to the scientific evidence we have at this moment. This is the end of this video. Thank you.